Hello, everyone. A very warm good afternoon to all of you. Welcome once again to your NEET Bytes here at Meritnation. And today we are going to talk about biology. So I hope so. All of you can able to see and hear me clearly. Hello, everyone. Once again. Good evening, good afternoon. So, guys, once again, we are here and we will continue our discussion from last class. That is your biological classification. So if you all remember, last time we were talking about living world and we did some of the questions related to that. Okay, and just subsequent to that topic, we have started with biological classification, and today we are going to talk about Monera in biological classification. And there's a small topic of virus that is there in your NCERT, and it's really important when you talk about your NEET. Okay, right. So let us quickly understand what are the different topics that we can do this time. Just a second, everyone. So all of you, biological classification. This is our topic for today. And hope so everyone can see and hear me. Now, so in biological classification, if you remember from your living world chapter also, we talked about a lot of classification in which many people have given their own sets of classification, starting from two kingdom classification, three kingdom classification, 4, 5, and 6, as well as 3 domain systems as well. So the most important of them all is your 5 kingdom classification, right? Though you need to know about 2 kingdom classification, who has given it, 3 kingdom, 4 kingdom, and why it was rejected, what are the problems in them. Okay, that you need to first you should know. Okay, and then you can understand that why your 5 kingdom classification is so much important and why it was very much taken into consideration when you actually classify organisms into different categories. Okay, right. Now let us move to our questions here. Okay, the very first question that I can show you is this one. Now, in 5 kingdom classification, everyone, if you can see here, okay. Okay, just give me a second. And so a 5 kingdom classification, if you can see, the main basis of classification is structure of nucleus, nutrition, structure of cell wall, and D, asexual reproduction. So quickly, give your answers, everyone. How many of you can give this answer? Okay, fine. I can find some of the, answers here. Okay, now most of you are saying in 5 kingdom system of classification, the main base of classification is B, nutrition, and that is absolutely correct. Now, why? So if you remember in 5 kingdom classification, everyone, the first classification was based on the cell and if it is a prokaryotic cell or if it is a eukaryotic cell. Okay, right. That was the very first basis. Right? Okay. Not the structure of the nucleus, not the structure of the cell wall, not asexual reproduction, but it was prokaryote and eukaryote. Fine. All right. And then we have divided them on the basis of autotrophic or heterotrophic nutrition. Right. And that is why your plant and animals are different into different kingdoms. And this is the reason. This is the reason out of all these 4 options, okay, B is the correct answer. Fine, all of you? Hope so. You have understood this question. So again, I'm repeating, for five kingdom classification, the very first thing that you should remember is that, first of all, it is classified on the basis of prokaryotes and eukaryotes. So prokaryote was given a separate kingdom, that is Monera. Okay, and all prokaryotes were unicellular organism or are unicellular organism. So basically, monerans are unicellular yet prokaryotes, and the rest four kingdoms. Okay, that is your Protista, your fungi, your plantae, and animalia are eukaryotic. Now, based on their food habits, okay, based on their nutrition, they have been divided into different. For example, if you talk about fungi, they are saprophytic. Okay, you talk about animals, they are heterotrophic, holozoic, right? Your plantae, they are autotrophic. So that is the reason, and that is the best possible classification we have right now of all the living organisms around this world or in this world. Okay, so let us move on to the next question. I hope so this is very much understood to all of you. Now, everyone, because we have got numerous type of classification, right? And the best one I will discuss after this question. So first of all, we'll do this question here. Phenetic classification is based on. Okay. So option number A, the ancestral lineage of existing organisms. B, observable characteristics of existing organisms. D, dendrograms based on DNA characteristics. And D, sexual characteristics. Okay, wonderful, wonderful. I'm getting the answers. I'm quickly getting the answers. It's an AIMS AIPMT 2003 question. Okay, and phenetic classification we are talking about, everyone. Okay, many people are giving their answers. Okay, and this time, okay, first of all, let us understand, everyone, the question. It is asking about phenetic classification. What do you understand by phenetic classification? So phenetic classification only takes into consideration of your external morphology, some of your characteristic. It never goes to your ancestral lineage. That is a phylogenetic classification. Do you remember this word, phylogenetic classification? Okay, there is a very important sentence also that we usually know in biology, that ontogeny repeats phylogeny. If you remember, yeah, exactly. If you remember, okay, when you were studying animal kingdom also, that in human embryo also, you will find pharyngeal gill slits. You will find a postanal tail, right? So it somehow captures your primitive characteristic when you are developing inside as an embryo, right? So that is the reason phylogeny, ontogeny repeats phylogeny, and phylogeny is the evolutionary characteristic. Phenetic classification has nothing to do with phylogenetic classification or ancestral classification. And that's the reason the correct answer is observable characteristic of existing organisms. Fine, all of you? Because no ancestral lineage is required in phenetic classification. Dendrograms. Now, let us see dendrograms. If you remember, that is your normal, tree. You make that different life tree or evolutionary tree that you make. Okay, that is called as dendrogram. So, hope so, everyone. You have understood this question. So you know the difference between a phenetic classification and also phylogenetic classification. Cool? Understood? Okay, great. Now, ready for the next question. Now, to which kingdom in the five kingdom classification do Chlamydomonas and Chlorella belong? Okay, this is a very easy question. Okay, A, Monera, B, Plantae, C, Protista, D, Fungi. But here people do a little mistake. So please give your answers quickly. Come on, everyone. I'm giving you 10 seconds. Think about it and give your answers. Okay, fine. I got the very first answer. That is C, Protista. Very good. Okay, C. People are giving answers as C. Very nice. So, okay, B also. Okay. First of all, Exactly. I've got one answer. Say they are algae. Okay. Absolutely right, everyone. It's not Protista. It's not Monera. It's not. First of all, it's not Monera because Chlamydomonas and Chlorella are eukaryotic organisms, right? No. Chlamydomonas and Chlorella are eukaryotic. They are not prokaryotic. So the very first answer, that is A, is now again, it's now clear. Okay, right? Then it cannot be fungi because they are autotrophic. Chlamydomonas and Chlorella both are autotrophic, and we don't know any of the fungi which are autotrophic. Okay, right? Now there are two options left: Plantae and Protista. Okay, Protista also have some organisms which can do photosynthesis. For example, Euglena, right? Exactly. But Chlamydomonas and Chlorella belongs to algae that is present in Plantae kingdom, right? Why algae? First of all, Chlamydomonas and Chlorella both have the chlorophyll A, okay, which is a characteristic feature of Plantae as well, right? Exactly, everyone. And they also have some accessory pigments, okay, which are typically of green algae. And we know algae belongs to kingdom Plantae. So yes, all of you, you are absolutely correct. The correct answer is B, Plantae, not C, Protista. So that's what I'm saying. It's a little tricky because people make mistakes between Plantae and Protista. Are you clear now, everyone? Cool. Great. So are we ready for the next question? Yes, we are. So here it is. Now, everyone, this is an interesting question. Okay, so choose the correct sequence of stages of growth curve for bacteria. Okay, and it has options as A, lag log stationary decline phase, B, lag log stationary phase, C, stationary lag log decline phase, and D, decline log and lag phase. Now, please let me first go through this question. This question you might not have seen, okay, in your biological classification, but it is related to the bacterial growth. Okay, that might you will study in 12th class when you'll study microbes. Okay, and in some places you will study in the Monera chapter as well. So this has a linkage from both the chapters, and that's why I have put this question so you people will understand that what are the different log phases, what are the different phases while the bacteria grow. So first of all, if you remember, when bacteria grows, yes, absolutely, you are giving very good answers. When a bacteria grows, it starts from a single cell, binary fission, okay, 2, 4, okay, 8, and then it keeps on growing. So at the beginning, the division is not very fast, but it has a good pace. This phase is called as lag phase. What it is called as lag phase. Here, if I'll just draw for all of you. So please, understand this question. So let's draw this graph like this. Okay, so the first phase, okay, will be as a lag phase. Okay, all right. Or I'll change the color of the uh, pen here, and so it will be very much understood. So this may be your lag phase. Okay, it was from 1 to 2, 2 to 4, 4 to 8, like that. Pace is very good. Fine, all right. But after certain divisions, okay, like for example, when it goes to 1 lakh, the count is 1 lakh. Fine, all right. After that division, after 1 lakh, it goes to 2 lakh, 2 to 4 lakh, 4 to 8 lakh. It is exponential. Okay, right, it is going exponential, and that's the reason, it's the log phase that happens. Fine, the log phase happens here, and the exponential growth you can see in bacteria. That's the reason. Okay, that's the reason. Overnight your food or maybe the curd can or the milk can convert into curd, right? It's because of the division of this kind of bacteria, right? Now, next is, see what happened. Every time where a bacteria grows, it requires nutrition, it requires space, right? It requires space. Now, what will happen? The nutrition comes to a to a quantity where now the bacteria starts growing slowly. Okay, so there is a stationary phase. Okay, fine. There is a stationary phase. Wonderful, all of you. You are understanding it very nicely. Okay, and now the nutrition is gone. The space is also very much full. Now there is no extra scope for growth or maybe division. Then bacteria starts dying, and then you have a decline phase. So these are the four phases. These are the four phases that you will find in the growth of the bacteria. Cool, everyone? The death that you can say. Absolutely right. Absolutely, everyone. Sriharin asked me that how is this graph different from plants? Okay, right. How this graph different from plants? First of all, in plant, you'll not have such a huge log phase, right? Their cell do not divide as like your microorganism, or maybe in this case bacteria. Okay, so there is a normal lag growth that happen. Okay, and they go to a stationary phase. That's the difference between the two. Hope you have understood. And yes, all of you, the very correct answer here is A. Got it, all of you? So this is a question that tells you about the reproduction of the bacteria or maybe any microorganism that divides by binary fission. Okay, come on, let's move further. Now, this is an interesting question, everyone. Thermococcus, Methanococcus, and Methanobacterium exemplify. It's an AIPMT 2008 question, and these are the few options here. Read the question, read the option, and then come to an answer. Fine, don't be in hurry. So I'll read the options for you one by one. So A is bacteria that contain a cytoskeleton and ribosome. Okay. B, archaebacteria that contain protein homologous to eukaryotic core histones. C, archaebacteria that lack any histone resembling those found in eukaryotes but whose DNA is negatively supercoiled. And D, bacteria whose DNA is relaxed or positively supercoiled but which have a cytoskeleton as well as mitochondria. Okay. Now, very interesting question. And people start giving answer. Okay, I've people are giving C as an answer. Okay. Okay, somebody gave B. Sriharin gave B. Varsha and movie maker, that's a very nice name, gave C answer. Okay, so most of you are giving C as an answer. Very good, very good, very good. Answer is C. Answer is C, everyone. Now let me explain you. Fine. If you find, first of all, when you read the question, you should have this in mind that what we are talking about. What type of organism we are talking about? Thermococcus, Methanococcus, Methanobacterium belongs to archaebacteria. Right, everyone? They belongs to archaebacteria. Now, all of you are in 12th class or maybe in 11th class as well. Okay, and in 12th class, when you study biotechnology, okay, and there is a small topic in biotechnology that's called as PCR, polymerase chain reaction. Okay, in polymerase chain reaction, there is a very beautiful enzyme that you know as tuck polymerase. I hope so. You know this name. Okay, tuck polymerase. Okay, T. aquaticus. That's the bacteria from which this particular enzyme comes from. Okay, and that particular bacteria belongs to archaebacteria group. Why? Because archaebacteria are those bacteria which can tolerate a huge range of temperature as well as acidity. Okay, so there are, absolutely right, you people are absolutely right. Thermus aquaticus is that particular bacteria. Okay, that's in short form I said T. aquaticus. Okay, no problem. Those who are in class 11, they will study in class 12. I'm just giving you an idea here. Now, so that means they have some property. They have some property, but yet archaebacteria belongs to kingdom Monera, right? We shouldn't forget about it. So the basic classification is that Archaebacteria belongs to kingdom Monera. And what we know about kingdom Monera, everyone, that they are eukaryotic, yet they are prokaryotes. Sorry, they are unicellular, yet they are prokaryotes. Fine. Okay. And if they are prokaryotes, first of all, there is no scope of having any organelle in them. So no matter if that's a mitochondria, okay, right? No matter if there is a cytoskeleton, okay, especially apart from flagella and all, right? Exactly. So they do not have such a good cytoskeleton or membrane system inside the body. Okay, except ribosome. Very good, very good. Okay, so that's the reason. That's the reason. The last option is wrong. Archaebacteria that contain protein homologous to eukaryotic core histones, no, they do not have. Okay, and bacteria that contain a cytoskeleton, it's an archaebacteria, so no, not at all. So the only correct answer here will be archaebacteria that lack any histones resembling those found in eukaryotes. Okay, but they're supercoiling. The way they actually, just supercoil around their proteins is positively. Okay, so this is a very good question asked in AIPMT 2008. It will clear your concepts about archaebacteria. Hope you have understood this. Okay. Now, next, move on, everyone. Now, this is another question which is quite related from your ecology also. Okay, quite related from your plant kingdom also. Okay, you will find this particular question in many different chapters. Okay, but it is also found here. Okay, so what is the question, everyone? A prokaryotic autotrophic nitrogen fixing symbiont found in. Okay, so first of all, we do understand what is symbiont. A symbiont is a relationship between two organisms, okay, which benefit each other. That's a symbiotic relationship, right? Exactly. And it says prokaryotic autotrophic nitrogen fixing. Okay, prokaryotic autotrophic nitrogen. Exactly. So the correct answer here. So what can you anyone can you tell me that which type of organism we are talking about? Because here it is the options which is giving Pisum, Alnus, Cycas, and Caesar. Okay, these are the plants. But what is that prokaryotic organism which lives inside one of this plant? Okay, as a symbiont. Can you name it? You people are right, absolutely, that it's C, Cycas. But inside the Cycas, which prokaryotic lives? Which the name? Quickly, I know you. You know the answer of this particular. Come on, everyone. Okay, fine. It's Anabaena. Not nostoc, not only bacteria, it's Anabaena. Fine, everyone. So Anabaena lives inside Cycas or with Cycas and have a very good symbiotic relationship between both of them. So Anabaena gives nitrogen to developing Cycas. Cycas give them food as well as shelter. So it's a very intrigued relationship. Okay, so this type of question you should know. Okay, it's quite a you need to learn it. I know, I understand that. Okay, okay. Nostoc are basically free living. Okay, they are not symbiotic. Okay. Here, everyone, now we have come to the virus part. Okay, now it's very easy question just to begin with. Okay, virus envelope is known as capsid, virion, nucleoprotein, or core. I believe that all of you should know the answer. And yes, I'm getting the correct answer very quickly. Fantastic, fantastic. Absolutely correct. You people are absolutely right. It's a very easy question. It's the capsid. Okay. And the best way to understand this is you visualize bacteriophage. Okay. Bacteriophage looks like a robot. Okay, it has a head, okay, have some tentacles like an octopus, okay, and the head is the capsid inside which the nucleic acid of that particular virus situated. Fine, everyone? Okay, so that's the question here. It's very easy question to begin with. Now I have another question for all of you, which is this. Which of the following biological phenomena involves a bacteriophage? Which of the following biological phenomena involves a bacteriophage? This is a combination of bacteria as well as virus part. Okay, this question belongs to both the categories or both the topics. Okay, if you understood, okay, and many people are giving, yeah, started giving answers. Very good, very good. Okay, so I'll start one by one, everyone. Okay, the very first one is A, transformation. Okay, those who are in 12th class, okay, they know what is transformation. They have studied this in genetic chapter also, genetic material when you were studying. Okay, it's a Griffith's experiment, very famous experiment, one of the most famous experiment of genetics chapter, right? Exactly. Transformation also you have studied in the sexual reproduction of bacteria, right? Exactly. So transformation never involves a virus. Okay, right? Never involves a virus, so transformation is out of question. Next is B, conjugation. It's a typical property of bacteria where it forms the pili, okay, and pass on its genetic material to another bacteria. That's conjugation. Fine, everyone? That's conjugation. Translocation is nowhere near, as a sexual reproduction of any of the bacteria or maybe, viruses. Okay, translocation is, you can say it's a part of mutation. Okay, fine, all right. And D is transduction. It is both involved with the help of bacteria and bacteriophage, where bacteriophage puts some of its, you can say genetic material inside the bacteria, and when bacteria burst and the bacteriophage which is formed later will pass on some of the genes of bacteria to another bacteria. That's transduction, which involves bacteriophage. Fine, all of you? So this is the question about bacteriophage, and you should have a knowledge of virus structure in the form of capsid. I have gone through the question, and also what are the different types of sexual reproduction of bacteria, where one of the type involves virus, that is transduction. Right, everyone? So hope so. Exactly. Hope so you have understood this particular session, okay, with respect to biological classification and Monera viruses. Okay, right. So I have completed these many questions. Now, what your job is to go and revise a little bit stuff. You know that where you are lacking now. Okay, just revise that part, do a few questions more. Okay, and let's meet up next Friday. Okay, and we'll continue this discussion of biological classification for other kingdoms as well. Fine. Thank you everyone for joining the classes. I'll meet you next time again, same place, same time. Thank you everyone. Take care.